Great speakers with Dr. Paul Wischmeyer and Dr. Craig Coopersmith. I'm Nazima Pathan. I'm based in Cambridge in the UK. My talk's going to be about the gut microbiome-host relationship in critical illness. Dr. Wischmeyer was then going to talk about nutritional modulations for the microbiome. And then finally, Dr. Coopersmith is going to discuss the gut as a source of organ dysfunction in critical illness. Some interesting data on an organ that's often forgotten in our management of critical illness. So I hope this provides some thought-provoking questions. As the session is being filmed, we'd be grateful to, if you have questions, to use the microphone so that the audience online can hear your questions before we respond. We thought probably we'd try and allow questions between each talk. Thank you very much. So, in terms of the gut microbiome in critical illness, I have no financial disclosures or patents. So what we do or what we don't do in critical care affects the health of the microbiome. A lot of the exposures our patients are undergoing, both as a part of their disease process as well as the drugs and lack of enteral feeding that they go through, that we put them through, are a risk to the viability of the healthy commensals in the gut microbiome. The problem is, I think it's becoming increasingly evident that the microbiome as a source of commensal species contribute enormously to our health, both locally but also systemically, as I'll go through. So I want to discuss how this matters and whether there, and then during this session we want to think about what capacity there is for recovery of the microbiome and how we can potentially support the microbiome which is undergoing tremendous change during the process of critical illness. So we live in synergy with a vast microbial community, both on our skin and on and within many organs of the body. And in particular with the gut microbiome, there's a number of important contributions to the health of the host, both immune, metabolic, endocrine, and digestive functions. In terms of immune, the commensal species and metabolites are produced, such as short-chain fatty acids, play roles in immune modulation with an anti-inflammatory activity, maturation of the lymphoid tissue within the gut mucosa, maintaining the mucosal barrier by helping the colonic mucosa produce mucin, which is an important layer to protect the sort of barrier between gut content and the blood system. And also by having a lot of commensal healthy pathogens, that helps to crowd out healthy organisms, that helps to crowd out potential pathogenic species in a state of health, and something that is lost if you're losing your good bacteria. Other important metabolic activities that the microbiome are involved with include metabolism of bile acids and lipids and biosynthesis of certain vitamins and amino acids. So again, these have implications if those bacteria are lost. Short-chain fatty acids produced by commensal species are important in regulation of gut hormone synthesis and which control gut motility, the feeling of satiety, as well as energy expenditure. And finally, of course, digestion of non-digestible polysaccharides results in production of these important chemicals and other fermentation activities that commensal organisms undertake. Apart from bacteria, what's increasingly clear is there is also an important population of viruses and fungi, all commensal and all beneficial, and these are being profiled increasingly as an important part of a community. And the microbiome isn't just these organisms, their metabolites are also important in terms of the organ system that is has such a deeply synergistic relationship with the host, receiving nutrients from us, but contributing to the health in the ways we've discussed. So talked about the fact that a lot of the unavoidable adverse influences in our patients, antibiotics, a vast majority of patients are receiving life-saving treatment with antibiotics, something that's going to be very hard to rationalize in the very early stages of critical illness, but as researchers have shown, that's likely to have a dramatic effect on the viability of commensal organisms. Other drugs also that affect the microbiome include opiates, the the process of ischemia reperfusion may be an issue in terms of the mucosa and the way it supports the healthy bacteria, hypoxia, lack of enteral feeding, and altered flow during bypass from a pulsatile to a continuous flow. These all may have an influence on the gut microbiome. And what we see are a range of things that are bad for the progression to multi-organ failure. Of course, we've lost commensals. Some of them may transform to virulent strains, and this may become important, particularly in those patients who have chronic disease, an expansion of the gene carriage of antibiotic resistance, damage to the intestinal mucosal barrier if we lose healthy bacteria and the production of mucin, loss of commensals, and also potential translocation of microbial antigens. So the gut barrier is an important way of compartmenting the microbiome from the host in health, and layers of that from the healthy commensal bacteria crowding out the potential pathogens to the chemical mucus layer and the physical barrier of the epithelium and the immune barrier of the mucosal-associated lymphoid tissue. They're all important and they're all damaged during the process of critical illness. If we briefly, I'll touch on intestinal barrier integrity, although Dr. Coopersmith is likely to mention this during his slides. There's a number of ways of looking at damage to the intestinal mucosa and biomarkers that have been looked at. In terms of epithelial damage, there's some circulating mediators that indicate the presence of epithelial damage, such as intestinal fatty acid binding protein, Claudin-3, which looks at the tight junctions between the mucosal epithelial cells. And then a marker of small intestinal injury is a measurement of serum diamine oxidase has been described as a biomarker. In terms of functionally assessing how well the barrier integrity is working, a lot of researchers have used dual sugar absorption using disaccharides and monosaccharides, which would not normally be absorbed, and measuring the 24-hour urinary excretion as an indicator of whether or not they are passing through and entering the bloodstream and being excreted in the urine. And finally, of course, the translocation of bacterial products such as endotoxin and a bacterial version, the bacterial isomer of lactate, which is D-lactate. These may indicate the presence of bacterial antigen translocation into the bloodstream if it's present. This is a rabbit model of hemorrhagic shock, and what this shows is how the normal colonic mucosa in health is damaged in hemorrhagic shock, so moderate and severe shock, and you can see a lot of damage and breakdown of the colonic mucosa in severe hemorrhagic shock, and that serum diamine oxidase is significantly increased. This is a paper published in 2014, and others have shown, but this animal model shows visually how a severe illness can affect the functional integrity of the mucosa. We had a look in children undergoing surgery for cardiac bypass, so a non-septic population, and we measured both a marker of intestinal injury, IFABP, intestinal fatty acid binding protein, alongside circulating endotoxin, and what we found was raised levels of these after congenital heart surgery. And interestingly, the endotoxin levels were correlated with inflammation, disease severity, inotrope score, metabolic acidosis as measured by lactate, and duration of intensive care stay. And other interesting thing was that we found in babies with duct-dependent cardiac lesions, those where we assumed the gut mucosa was underperfused preoperatively, they had higher amount of postoperative endotoxemia and mucosal injury, so they were particularly susceptible to the process of cardiac surgery. So we've talked about how the gut contents are disturbed during the process of critical illness, and the way we look at this and have been describing this is in terms of the density, because normally there's a very dense, diverse population of bacteria, and we have a certain sort of expectation of healthy commensals, which change through life from a very sparse population in newborns and babies, and then the maturation as feeding progresses into the diverse population we see in adults. How do we measure this? Culture-based methods are not effective because they miss unculturable and low-prevalence species. Sequencing technologies have started to look at this in critical illness. Fecal samples are easy a way of accessing and assessing lower GI microbiome, although potentially not truly representative of the colonic microbiome, but it's the best we have. Rectal swabs are another way that researchers have looked at sequencing the gut microbiome as well. So I've talked about how we've got expansion of antibiotic resistance, gene carriage, but also now we're going to look at what studies have described in terms of the intestinal microbiota. So this is a study by Lankelmer, and this was where they looked at 30 ICU patients and 15 healthy controls. And they profiled, they did rectal swabs on admission to the ICU. What you can see in the healthy patients is a fairly uniform fairly uniform sort of picture, although there's some variation, but lots of firmicutes. This is phyla level data, lots of firmicutes and bacteroid, but sort of relatively stable population of mainly bacteroids and firmicutes. But in the critically ill patients, you can see there's quite a bit of variation. And what these authors found was a significant interpersonal variation and expansion of certain phyla associated with pathogenic species, such as proteobacteria, and a loss of commensals. This has been confirmed. Dr. Wischmeyer's group has done some work with a large number of patients, of critically ill adults, comparing it to data from the Healthy American Genome Project, healthy healthy individuals. And what, again, you can see is that the proportion of bacteroids and firmicutes are depleted and an expansion in proteobacteria and potential pathogenic species. In this study that Michael Morowitz's group did, this is looking at a number of sites in a group of critically ill children. So 27 critically ill children, and they compared them to 30 healthy children. The mean ages were slightly older in the healthy children, and I think that's slightly relevant because the microbiome is age dependent, and they also did compare it to data from the Human Microbiome project of 277 adults. So this is samples they took of rectal swabs or fecal samples for the GI, skin microbiome and tongue microbiome. And what you can see in healthy adults is the first column, and then you've got healthy children and a sort of classic bacteroids and firmicutes, and this is the healthy child skin microbiome, relatively low, and this is the healthy child tongue microbiome. And then if you look at this sort of profile of what the patients in the PICU are demonstrating, you can see a loss of commensals and a huge number of other species filling in the gaps. And that is what is of interest, because that is potentially contributing to the severity of illness in these children. We've had a look similarly in age-matched critically ill and healthy children, and you can see here that a sort of the pink is the controls, and they're fairly well clustered, and then you've got a sort of quite a scattering of children, of critically ill children. And what this does, this is a Pca plot, and you can see separation, how the overall sample profile compares with each other. And this is looking at diversity and richness in the healthy children and the critically ill children, and you can see a reduction in diversity and richness of the fecal microbiome. Then we did some machine learning to look at which species amongst those that are significantly different are driving that separation, and you can see that in healthy children you've got lots of commensal bacteroids, Faecalibacterium, and that in the patients the Enterococcus prevalence is particularly important, and that seems to be driving the separation, and that's what's really expanded in our critically ill children. So many authors have looked and shown that the composition of the microbiome has changed in critical illness in adults, in children, and there's been work in neonates as well. How is this clinically relevant? How does this actually make a difference? Oops, sorry. So Lankelmer's work, that I showed earlier, where they showed hyper-individual variability, they looked at sort of bacteroids Firmicutes ratio. They had seen an upregulation in the number of gram-negative species in patients compared to controls, and also saw an increase in an increase in Enterococcus, but they did not see any association with outcome in terms of sepsis or death or other adverse events. This group, Dr. Friedberg's group, looked at 300 critically ill patients. They cultured and did rectal swabs and looked at whether prevalence of certain species was associated with sepsis or death. And what they found is that the presence of VRE on culture, vancomycin-resistant Enterococcus, was associated then with VRE sepsis, presence of E. coli, Clostridium, Pseudomonas, and I can't read it very well. Thank you very much, Klebsiella. They were associated with sepsis with that particular bacteria. This is on the fecal sample, and then they've got blood sepsis. They didn't, interestingly, see anything with Enterococcus. A lot of patients had Enterococcus, and they classified patients who had more than 30% Enterococcus. That was called Enterococcus dominance. They didn't see an association with Enterococcus sepsis, but what they found was that patients who had Enterococcus in high amounts or VRE colonization had a higher risk of death or all-cause infection. So clearly, Enterococcus is bad in this group. Both others and we have also found this. Okay, so we know now that microbiome depletion, you're losing the good guys, and you've got lots more of these pathogenic species. What is the consequence of losing the functionality of those healthy bacteria? So to look at what those bacteria are producing, researchers started looking at metabolic profiling. This is an early paper in 2006, and both in this group, Shimitsu's group, has looked both in adult patients with SIRS, systemic inflammatory response, trauma, and critical illness, and consistently finding that short-chain fatty acids, those very important metabolites that contribute to our health in terms of immune modulation, maintenance of the barrier, they are massively depleted compared to healthy adults, and particularly butyrate. You can see here from a sort of level of 16.6 down to undetectable levels in patients with SIRS. And we have also done some work looking at metabolic profiling in children who are critically ill. This is a sort of a plot from NMR, and this is our patients. We compared age-matched healthy children and critically ill children. And what this shows, these peaks, the ones that are significant are the ones I've labelled here, and you can see butyrate, acetate, and caprolate are short-chain fatty acids. They're present in the controls, and they're absent in the patients. And what we're seeing is complete loss of short-chain fatty acids in critically ill children, just as in the adults. And our data and the work published by Shimitsu's group have shown that SCFA levels, when you lose them, the levels are associated with higher inflammation, higher risk of sepsis, higher mortality, high risk of inotrope requirement, and a longer duration of mechanical ventilation. So clearly, not just locally, but overall disease severity, there is an association. So that's one thing. In the slides previously, I also talked about the fact that those healthy bacteria have many other functions, and one of the ones that I'm interested in is looking at bile acid metabolism. There isn't really very much out there. This study and others have looked at the effect of antibiotics and how that, because pretty much all of our patients get antibiotics. I'm a pediatrician, so I'm not, but looking, my reading of the adult data is about 70% of adults get systemic antibiotics, and in some units using selective digestive decontamination, all of their patients are getting it. In our population, again, pretty much 80 to 100% of children at some point during their ICU stay receive antibiotics. And so the bile acid pool is affected. And what this slide shows, and this is in a rat model receiving antibiotics, the red is what's up and the blue is what's down. And essentially what this data is showing is at the top here is different bile acids. And what we're interested in is that in the in the rats treated with antibiotics, we have an upregulation an accumulation of primary bile acids that your own metabolism generates, and a downregulation in the abundance of secondary bile acids, which is what we depend on commensal species to generate: Bacteroids, Clostridium, Lactobacillus, Eubacterium, Bifidobacterium, Listeria. Loss of these bacteria means we're not producing secondary bile acids. And again, some pilot data from my group showing that in critically ill children, the primary bile acids, so cholic acid and taurocholic acid, are high in the patients compared to controls, and the secondary bile acids, deoxycholic and lithocholic, and many others dependent on the presence of commensal species, are reduced in the fecal samples. So I hope what this is showing you is that loss of commensal species and accumulation of pathogenic species, and that disruption to the healthy balance between gut and microbiome and the host has a potential risk to other organ systems. And in the ICU, the long-term risks are not defined of this. There's lots of evidence that the gut, through its a deep lymphatic and blood and nervous connections to the brain, the heart, the lungs, the kidneys, has association with many diseases. And so potential is that we do need to think about this, if not for the acute disease severity, then for the longer term risks. And I know in neonates that there is a risk between early antibiotic treatment, later risk of atopy and asthma in those children who've gone through the NICU. So there is a risk, and this health and wellness could be something we could modify for our patients' benefit in the longer term. What can we do to prevent gut failure? I think Dr. Wischmeyer is going to talk about this, and I think he'll agree with me that the solution needs to be probably quite personalised, because as our data and others has shown, there's a huge variation dependent on what antibiotic, what feeding regime, and whether there's other comorbidities that are contributing to the type of dysbiosis in our individual patients. Oops, thank you very much. And if you have any questions, please let me know. Thanks. Is there any questions at all? No? Okay. I'll introduce Dr. Wischmeyer. Thank you very much.
Wait for my slides here for just a moment. So what I hope to take further is the concept that we spend a lot of time in the ICU and in other settings worried about how to eradicate the bacteria that live in our bodies and the microbiome that perhaps lives in our bodies because we fear its infectious capacity. But what I hope to impress upon you today is perhaps instead of removing and eradicating bacterial species, we should be intervening to give back if my clicker will work. Maybe it won't. Can I advance? There we go. To treat patients, we should be giving back, perhaps, bacteria and perhaps even stool to our patients. So these are my alignments of interest. I'd like to believe that we're not in conflict with the funders we work with, but in alignment with the funders we work with for better patient outcomes. And so what I hope to impress upon you is that realistically, our microbiome and our bacteria that live in our bodies are not enemies, but 100 trillion friends we didn't know we had, and perhaps friends we want to keep around. And what I'll focus on is give back, and give back to our patients. And this is already happening. Many of you are probably aware that we are already starting to prescribe stool, bacteria, and poop pills to our patients. There are books about it. You can do this at home in your blender. I might recommend against it, but it is still possible to read about. And the day is coming when we will write prescriptions for bacteria rather than prescriptions for antibiotics to remove bacteria. And of course, many of you probably know we're doing this already. This is just one of the many stories of patients whose lives have been saved by the power of poop. This is a young woman named Caitlin who was in a car accident in the Southeast, and 30 days into her stay was ready to go home but got C. diff and went through 9 rounds of antibiotic treatment, was dying of a toxic megacolon when she got a fecal transplant from her mother. Immediately, she recovered from her C. diff, never to have it return, and went on to recover and go home very quickly thereafter. So already we're realizing this can save, lives. Can I have it back? maybe. Can you help me in the back? What just happened? Yeah, maybe so. I think that's about all I get, Craig, eh? Can you pull me to the top, please? I need to go all the way to the top, first slide. Go back in. No, don't do it that way. Go out. Go out. Pull this cursor right here. Let me do it. Pull this to the top. Or actually, I can't click on it, can I? Yeah, I can. Oh, this is pretty neat. Okay, now let's go back to where we were. Okay, play. Beautiful. So why should you care about your microbes? Moving on. Well, have you ever wondered why when you go into the woods, you get bitten by all the mosquitoes, but your wife, husband, girlfriend, boyfriend does not? It's not because you're sweeter. It's because the microbiome that lives on your skin secretes chemicals that attract or repel mosquitoes. So it's actually your bacteria that are sweeter or not sweet. And in fact, we're discovering the microbiome affects many things in our lives, including perhaps who we have sex with and who we marry. At least we know that's true if you're a fruit fly. So perhaps we'll find out it's true for humans as well, but we aren't quite there yet. But it should make all of us a little nervous. So how do they do all that? And it begs the question of what really makes us human. And some of us think, well, perhaps our cells make us human. Well, we have 30 trillion human cells in all of us sitting here right here today, 38 trillion microbial cells. So cellularly, we are more bacterial than human. Perhaps you think it's your genes that make you human. Well, you have 20,000 human genes and 20 million microbial genes, so you're 1% human genetically and 99% bacterial. Something to think about. So we are finally poised, as you heard in the first lecture, to begin to answer questions, and you're seeing data presented finally that begins to look at this using microbiome sequencing, not just to describe who we are, but describe how we can care for people better and what happens. So the real question is, can we use the microbiome to cure and treat disease? Well, we've already showed you that there are patients already getting this intervention to cure and treat disease. But what about what's in our patients' guts? Can it help us? Can it hurt us? And can we change it? Well, we know that what's in our guts changes dramatically. We've already begun to see data. And even stress, the stressors in our lives, who our president is, the driving on the busy roads affects our microbiome, and our microbiome affects our brain. Microbiome secretes neurotransmitters called psychobiotics that affect depression and anxiety, and our depression and anxiety changes the microbiome of our guts, and so it's important to realize it's a synergism. And as you heard before, all the things we do in the ICU affect the microbiome as well. I was a disciple and trainee of John Alverdi, who taught me even the opiates we give cause our microbiome to become more virulent and more aggressive, but clearly the antibiotics are a major offender. And so the reality is perhaps we need to be restoring balance with probiotics, poop pills, fecal transplants to resod the lawn that we're blighting through the stress of critical illness and through the antibiotics we administer. In fact, I can tell you the students at MIT will sell you their freeze-dried poop and tell you that it will make you smarter. The FDA has some problems with that, but it's something to think about. So the probiotic literature is exploding. This is not even up to date. It's going up even faster. They are in all of our foods. They're in many of the things we eat. They are everywhere. There are books on this as well, by the way, if you want to read more about it. But if we're going to use a probiotic, a perhaps live organism, we should be thoughtful about it. It should make scientific and mechanistic sense. And so the science needs to be there. And so we have a growing science. This is work that John Alverdi and Gene Chang, my mentors in the world, many years back at University of Chicago did. And essentially what we've learned is when the body becomes stressed or we become ill, our normal commensal flora disappear. And we'll show you data, and you've seen data for that. It's a little bit like when there's a hurricane in New Orleans, all the regular people leave and the looters come and they bring their rowdy friends. That's exactly what's happening in our patients' guts. When the normal bacteria leave, it's the pathogens that show up and they bring their rowdy friends and they start looting. And the way to make them go away, perhaps, is to bring the regular folks back to town. And that's the idea of probiotics. So it doesn't really matter, we believe, what probiotic you give, we're discovering. Anything will outgrow C. diff. C. diff only grows when everybody else has left, much like the looters. And so the idea may be, although it's nice to target probiotics, and I'll give you some targets in a minute, any normal commensal may do the job. So prebiotics also play a role. The fructooligosaccharides that are in many of your tube feed formulas and other fibers play a key role, and they get generated, or they get turned into the butyrate and the short-chain fatty acids you heard in the last talk play such a key role in outcome. But clearly, the lack of them then lead to much more aggressive bacterial species that then can invade the gut. And Craig will, I'm sure, tell you the master's version of what this looks like. But this is a brilliant paper in Nature Immunology recently that really is enlightened and has some beautiful figures in it that talk about the role of probiotics and prebiotics and the microbiome in inflammation. So there's many mechanisms by which probiotics may protect the gut, and these are just a few of them that my lab and other labs have been fascinated by. So clearly, some of the things we're really interested in is how to make this science clearer. So we've modeled this, and Craig will probably show you some of this data too, but this is work actually that some of the people that used to work in his lab did, looking at probiotics and how they affect survival, in this case, peritonitis and Lactobacillus GG, the culture oil you can buy in the store, and no, I don't work for them, and bifidobacterium used in the NICUs around the country and around the world. Both can reduce mortality from experimental peritonitis and from experimental pneumonia. And I won't show you that data. And they perhaps do this by reducing apoptosis in the colon. You can see much less apoptotic cells in the 2 probiotic-treated animals. And if you look here, this is the blood cultures from an animal with peritonitis. This is the unprobiotic or non-probiotic treated animal. There's a smear of bacteria. There's severe bacteremia in this animal. This is the same time point in an animal that's been given LGG. None. I made Lita, who Craig knows well, repeat this experiment 15 times because I didn't believe it. But it's been compelling, and we've been able to show it for pneumonia as well. So there's a barrier function and an immune function through mucins, through Treg cells that probiotics we've shown can affect. Basic science is a start, but it's not sufficient. We need clinical trials, and we need safety, right? We're giving a live organism, and lots of eyebrows get raised when you start talking about giving bacteria to patients. In fact, the government thought for sure this was going to cause harm, and they spent millions of dollars with the AHRQ to look for harm in probiotic treatment. And they published a 645-page treatise to show you there was going to be harm. They looked at 622 trials, every human trial ever performed with probiotics, and they did not find it. They could not find any harm from any probiotic, save one I'll tell you about, intervention. And in fact, relative risk of 1, believe it or not, people who were acutely ill, critically ill, or more sick actually had a tendency to benefit from a probiotic intervention versus have risk. There was an exception to that, the Pca trial. This was a probiotic preparation given to pancreatitis patients with fiber post-pylorically. Anyone have an issue? Think of why post-pyloric administration of anything to a pancreatitis patient might be troublesome if it sat there. Well, unfortunately, they broke their stopping rules when they saw a mortality difference, thinking the probiotics were helping, and they kept enrolling, and the mortality difference got bigger. Unfortunately, it wasn't favorable to the probiotic group. Actually, what was happening was the fiber was sitting just beyond the feeding tube, causing a nidus for bacterial growth. Lack of motility led to ischemic bowel. I actually talked to the surgeons who operated on these patients. They would find niduses of fiber sitting beyond the feeding tube and ischemic bowel beyond it. And so, rule number 1, don't give probiotics post-pylorically, because the stomach will take care of them if the motility is poor, the small bowel will not. This is the only trial that showed any harm in any of the trials of probiotics ever in the literature. Unfortunately, it had all kinds of ethical issues as well. So in all 622 trials examined, this was the only one that showed a safety risk, and it taught us a lot. And so they found and concluded that probiotics do not pose a risk and perhaps are beneficial to the more acutely ill patient. So what's the clinical data, the published clinical data for outcome? Well, let's start big first. This is the big meta-analysis in JAMA looking at the effects of probiotics on antibiotic-associated diarrhea. About 12,000 patients, 63 trials, reduced antibiotic-associated diarrhea 40%. So a big effect, 40% absolute reduction. This is a trial that Lee Morrow did with an NIHK award, actually, looking at LGG, Culturelle, again, the stuff you can buy at the grocery store, on patients who were intubated, smearing 1 capsule in the mouth, putting 1 down the feeding tube, and their incidence of VAP. Reduced ventilator-associated pneumonia by half, and that was true in the intention to treat group as well. So a significant effect in this population. The group I work with has meta-analyzed this data. This is one of the more recent meta-analyses. There will continue to be more, I'm sure. We saw in 30 trials of 3,000 critically ill patients, significant reductions in infections, VAP, and the sicker you were, the more likely it was to help you. Again, the more deranged perhaps your microbiome is. This is the table. It was a 20% reduction in infections in this population. There was a lot of heterogeneity. Not one of these trials used the same probiotic at the same dose in any of them. So again, Rowdy friends get knocked out by normal people showing up. All of these seem to have a benefit. Some may be better than others, we'll find. This is also true. There's meta-analyses in elective surgery and trauma and other conditions we treat in the ICU. I won't spend time covering them, just safe to know the literature's out there. There's also a meta-analysis for depression and the role of probiotics in depression being beneficial. So it's not just for the immune system and the metabolic system. But this is the landmark trial. This is the trial which by all other trials in probiotic and prebiotic therapy will be measured. It was published in Nature last year. Pinaki Panagrathi spent 10 years of his life and millions of NIH dollars studying 4,500 normal full-term infants in India. He randomized them to Lactobacillus plantarum with fructooligosaccharides, the fiber that's in many of our tube feeds, versus a control, versus placebo. He significantly reduced sepsis and respiratory infections in full-term infants in this population. This was a dramatic effect. This was published in Nature, not Nature Medicine or Nature this or that, 5 pages in Real Nature. So this is the trial by which all others will be measured. Had a significant reduction in the primary outcome of lower respiratory tract infections and sepsis, 40% reduction in the largest trial ever conducted of a probiotic-prebiotic therapy. In fact, he attributes it to the Treg cell data that Lita and people from Craig's lab who came to work for me published themselves on why the respiratory tract is so affected by the gut. We need large trials like this in the critical care unit. We need definitive trials targeting populations. We kind of thought maybe we were there with C. diff. What about probiotics and C. diff? This has got stool prescriptions written left and right. Probiotics are effective here as well. Cochrane analysis shows 64% reduction in C. diff. This doesn't seem to matter whether this is a child or an adult. Doesn't matter what species you give. It seems to work in different doses. Again, C. diff is the most pathetic organism in the world. It will grow in isolation wonderfully. It will not grow in the presence of other commensal organisms, is what we found. You can actually see this in action. This is the University of Minnesota's data. They actually took a number of C. diff patients. Again, those are stool samples, those stars. They don't look anything like normal stool samples. And they gave a stool transplant to a number of these, the donors down there. Watch closely to what happens within 24 hours to the stool samples of the patients when they get a stool transplant. They return to normal and they stay there for weeks. These are replicate samples of patients and their microbiome appearance. So again, we know that we can correct rapidly with fecal transplants and restore balance to these patients. So where do we go from here? Large trials of probiotics, stool transplants, poop pills for everyone. Darren Hyland, Deb Cook, and I took a LGG trial to the NIH, and we were told, much like the TV doctors know, one probiotic is not likely to fit all. And so they asked us to go back to the drawing board because one size is not likely to fit all, and tell them what was missing in the microbiome of critically ill patients and come back to them with targeted trials. What should be in the poop pills is what they wanted to know. How will we define that? And so can we use the microbiome to do that? And that's some of the data you saw. The last talk presented, that's what led us to do the ICU Microbiome Project. It was to target a probiotic trial. This was our first publication. You saw it before, 115 patients. You can't be in an ICU in North America, these were Canadian sites as well, and not get antibiotics if you're there 48 hours. 100% got antibiotics. We couldn't find a patient that didn't. We compared them against 1,200 patients in the American Gut Project. Rob Knights made $2 million sequencing 6,000 citizen scientists. Human Microbiome Project cost $173 million to synthesize 200 people. That's crowdfunding in action. Fecal microbiome is largely different. The blue there is the fecal microbiome samples. You can see whole families of normal bacteria lost. The white dots are the ICU patients there. White means no Firmicutes species at all, so whole families are being depleted within days. Red is the normals, healthy subjects on that side. This should scare you a little bit. Your stool, your mouth, and your skin begin to look the same. You lose diversity in all sites. We think this is loss of barrier function, but your stool and your mouth shouldn't look the same. But they do in ICU patients. And these are, again, the diversity plots you saw before. Large amounts of proteobacteria, which are the pathogens dominating the ICU patient versus healthy controls. We found targets. There's huge depletions, and you saw in Nazima's lecture as well, the same Faecalibacterium that makes short-chain fatty acids and is a major target in IBD, massively depleted in the ICU. This is a target. It's an anaerobe. It's hard to give. We're growing it in my lab right now in anaerobic chambers, but this is a target. Huge overgrowth of pathogens. We actually showed there's association with death with Klebsiella on your skin, association with bacteremia, with mycoplasma in your mouth. But you can really see this looks a lot like the plots you were showing. Huge Enterococcus staph overgrowths, large losses of Faecalibacterium. So these are our targets, and they're the same targets the IBD doctors have known for 10 years. We're just always a little behind. So these are the future probiotic targets we are after. We have lots of data we haven't published from, and if you have questions or want to get into this field, we're happy to share with you. Rob Knight is a very proprietary sharer, and this data is publicly accessible. Some ongoing projects we're doing that are critical care related. We're collecting microbiome samples in Everest with the Everest Extreme project. We actually have these samples already, looking at hypoxia's effects on Sherpas and on European climbers up and down the mountain from peak to bottom. So we're looking forward to analyzing those and seeing what hypoxia does to us. And then the chair of surgery, Alan Kirk, who used to sit next to Craig at Emory, has put $10 million into sequencing all omics, immune outcomes and clinical outcomes in 1,000 consecutive surgery patients. So soon we'll have all this data merged together, courtesy of the good Dr. Kirk and surgery. So in closing, what have we learned so far? A healthy gut is a diverse gut. Unfortunately, ICU leads to a massive loss of this associated with a poor outcome. And we need to find ways to correct this, and we believe probiotics and poop pills may be one of them. And if so, we can change the world we live in, the microbial world, since we are mostly microbial, and use the power of poop, perhaps, to change the outcome of our patients in the future and perhaps resod the lawn that is blighted by what we do to patients in the ICU. And this affects not only the infection rates, the lung, and the things we've talked about, but again, anxiety, pain, and many of the psychologic effects that the neurotransmitters our microbiome secretes affect as well. And we have trials that have shown this in large scale. We only now need to take our lessons from trials like this and do similar trials in our patients to restore the 100 trillion friends we didn't know we needed, restore balance to our patients, and now make it so easy a child can do it using our new techniques. And so you can get involved too. Feel free to send your stool to the American Gut Project. You can sequence your stool, your dog's stool, your wife's stool, or sponsor a child in Africa's stool. This is who analyzed all the data from our ICU Microbiome Project, Daniel, but we need more patients. So we take the kids into the lab to show them how the robots analyze the stool. They're not always so thrilled, but we think it's pretty cool and sexy and exciting. So I hope you do too. Thanks to Rob. This is some sampling of Komodo dragon at the Denver Zoo. And we'll look forward to your questions after Craig Coopersmith gives us his talk on protecting the gut and gut failure.
Did anybody notice in the picture of McDreamy just how much it looks like Paul Wischmeyer? Just saying. Okay, so I'm going to give everybody the capacity to leave right now because we sort of did this backwards. So they talked about clinical data, and I'm going to take us directly from clinical data into mice. And I'm going to talk exclusively, I'm going to probably skip over for time all the couple of clinical slides I have and go directly into mice to actually explain some of the mechanisms. So rush for the door, it's okay, I'm cool with it.
Don't rush for the door, it's the best part of the talk.
So my disclosures are here, and none of them are even remotely relevant to what I'm going to talk about today, except for the fact that my lab is funded by the NIH. So this has briefly been talked about. I'm not going to exclusively talk about the microbiome. We'll see how I do on time. But the gut is actually made up of three parts. The gut is made up of an epithelium, which depending on who you ask is somewhere between 30 to 300 square meters, which is the size of a tennis court or half the size of a badminton court. Show of hands for who knows how big a badminton court is. It's the size of your gut epithelium. There is an adaptive immune system. The adaptive immune system, a lot of people don't know this. There's more lymphocytes inside your gut than any other place in your body. Not the spleen, not the blood, the gut. And I'd feel like it would be ridiculous for me to talk more about the microbiome since we've just heard 40 minutes of talks on it. So historically, the gut has been called, quote unquote, the motor of sepsis, but our understanding about why gut might be the motor of sepsis has evolved fairly remarkably over the last 30 years. Not going to talk about that. So real quickly, briefly, clinically, how do we measure gut failure in the ICU? We measure it remarkably simply. No bowel sounds, diarrhea, bowel distension, vomiting, high residuals, GI bleeding, intra-abdominal hypertension. Who here thinks that any of these correlate with death? They don't. But if you put them together, they do. So the number of simultaneously GI symptoms are higher in non-survivors than survivors. And gastrointestinal failure, defined as 3 or more of those that I just showed you, all simple things, but 3 or more of them on day 1 independently is associated with a 3-fold increase in mortality. I'm going to fairly rapidly go from association to causation, which is why I do the nerdy mouse stuff. So are there any more biological markers of gut failure? Again, this is the last human data I'm going to show you. So the first speaker talked about a little bit of this. Plasma citrulline you can measure as a marker of enterocyte mass, and plasma IFABP, or intestinal fatty acid binding protein, can be measured as a marker of an enterocyte damage. So you would think if your gut is damaged, what would you see? Less mass, more damage. And that's actually what you see. So this was published last year or 2 years ago in Current Opinions in Critical Care. It's sepsis, it's CPR, it's critical illness, it's everything. And what you see simplistically is less mass, that's less citrulline, and more damage, that's more IFABP. So mechanistically, how can the gut serve as the motor of MODS? Well, there's about 15 different ways, and I'm going to talk, I think, about 3 of them. Paul already showed this data, and Paul did this data. I didn't. But it's just to show you really simply, red is us, blue is our patients. This is not a political statement, not red state, blue state. But red is us, blue is our patients, and we look different, entirely different. That's the last thing I'm going to talk about clinically. So before I actually talk about this slide, almost everything that was shown to you in the last 40 minutes in my first 3 minutes and 38 seconds has been associative. We know that the microbiome is different in our patients. We know our patients die. We know there's an association. We know that we can do studies of the microbiome in stool transplant, et cetera, in healthy patients and recurrent C. diff. Almost none of that has anything to do with the patients we take care of in the ICU on a day-to-day basis. And so I'm going to try to move you a little bit now from association to actually hopefully proving causation. So in order to do that, we can't until we have positive clinical trials in the ICU show that. So everything I'm going to talk to you about for the last 16 minutes is going to be in mice. Again, feel free to run for the doors. The first study, and this was studied, this was published about 5 years ago by Paul's mentor, John Alverdi, and this was published in the journal Surgery, and not too many people know about it except for people who've heard me talk. And I think this is debatably the coolest study I have seen in the last decade. So I'm going to take you through this, and I'm going to take you through it slowly. You're welcome to read what it says on the slide. Take Pseudomonas, inject it into the cecums of healthy mice. Take that Pseudomonas out, put it into a different mouse. All the mice live. Take Pseudomonas, put it into the cecum of a mouse that's undergone a 30% hepatectomy. Inflammation, not fatal, but inflammation. Take that Pseudomonas out, put it into a different mouse. All the mice die. Bacteria sense inflammation and badness inside of us. They change their virulence factors, and they become the looters and the marauders that Paul was talking about. The implications of this are remarkably profound. Same bacteria. The bacteria says, today, I'm friends with you. Tomorrow, you're my enemy, and I'm going to come at you with something that I'm going to try to kill you. So this totally puts on its surface we should give antibiotics to everybody or change it, because what's happening is bacteria are changing their virulence factors. So the implications of this are remarkably profound. It's not just giving antibiotics. Virulent bacteria can cause sepsis and remote organ failure without systemic dissemination. The next number of slides, probably the next 8 or 10 slides I'm going to show you, are all unpublished and currently under review from my group. So if you take pictures, please don't share them. I understand this is live streaming, so maybe I shouldn't show them at all. But, because, I hope. We take genetically identical mice from different vendors. Beautiful thing about mouse studies is you can literally make them genetically the same. And if you buy them from different vendors, the assumption is they're the same. We can make them the same gender if we want to. We can make them the same genetic background. We can make them the same age. We can make them the same everything. But if we buy them from different places, the only difference is their microbiome. So we did that. We took age and gender matched genetically identical mice from different vendors, and I'll explain why, but we did females because when we do some experiments, I'll explain later, if you put male mice in the same cage, they fight, and if you put female mice in the same cage, they don't. This is not a sociological statement. It's a factual statement about mouse biology. So we take mice from different vendors. We make them septic by cecoligation and puncture. These are the different vendors, and we see a marked difference in mortality. Again, the only difference between these mice the only difference is their microbiome. That is still an association. This is a little bit more data looking at alpha diversity, and that's within group, within a certain mouse. You see that one group, the red group, again, not political, but the red group here, that's the one that had higher survival, has higher diversity within it. Again, we already heard about an association with diversity and outcome. This is beta diversity. This is between samples. And this is just showing you that, in fact, the groups are different. They're different at baseline, and they're different after you make them septic. And although I have about 1,000 slides, I'm only going to show one because, again, we haven't actually published this yet. It affects the immune system. So there's an association. Again, everything I've shown you so far is an association. You change the microbiome, you make the animal septic, while the immune system is the same at baseline. That's the first two columns. Once you make the animal septic, the immune system is different. This is specifically looking at effector memory CD4 cells, although again, we have lots and lots of data to suggest that the immune systems, once you make them septic, is different. So hopefully I'm going to now move from association to causation. So we have genetically identical mice from different vendors. They have different survival, and that's associated with different microbiomes and different immunophenotypes. What happens if we take genetically identical mice from different vendors and we give them a common microbiome? How can we do that? We put them together. So again, this is where we can only put females from different vendors in the same cage. If we put males from different vendors in the same cage, they will fight. So even though the NIH for reproducibility wants to use both genders, we can't do this in male mice because they'll kill each other. But if you take female mice from different vendors, they will get along perfectly fine. And so you could take female mice from different vendor, from vendor A and vendor B. The only difference is the microbiome. You could put them together in a cage. 3 weeks later, assess their microbiome. Why? Because mice, unlike us, eat their own stool. And so they would eat each other's stools, whether it sounds gross or not, they eat each other's stools, and ultimately, after 3 weeks, they will develop a common microbiome. So they start with different microbiomes, and they start with different mortality. You put them together, no intervention, just let them eat each other's stool, and what happens? Their diversity, remember I showed you there was a difference in the diversity? Their diversity change totally goes away. The between group variance totally goes away. This is a little bit of a complicated one, but I'm going to ask you to look at colors. We're going to look first at the left and the right. The left and the right are the pre-co-housed. I hope you will see that there are different colors. The one on the right has a lot of brown and pink up top and is less diverse. The one on the left is more diverse. You put them together, you let them eat each other's stool, that's the middle two, and it doesn't matter which the origin was. They end up looking the same, and they end up looking like the diverse one. They have, by eating each other's stool, they've come up with the same microbiome. And now we do survival. And what happens when we do survival? The ones that are co-housed have the same survival better than the one that previously had a less diverse thing. This mechanistically proves that by changing the microbiome, we are changing survival. I'm not going to show any data on it right now, but all the changes in immunophenotype, again, I only showed you one, all the changes in immunophenotype go away by developing a common microbiome. So that's all I'm going to say about the microbiome, because when we talk about the gut as the motor of critical illness, it's not just the microbiome. So there's like 30 years worth of data on this, and all the microbiome data goes back 6 years. So I'm going to talk about two other things in the 9 minutes I have left. So our first speaker talked briefly about permeability. So the intestinal barrier function. The intestinal barrier is, there's tight junctions that allow things to move back and forth. And so it is not a total barrier. So the things that live inside the lumen, some of them can get out. Some of the things that live outside can get in, but it's regulated. It's regulated to allow movement of water and solutes and immune cells. And it's modulated by tight junctions. But tight junctions are compromised in critical illness, which leads to increased permeability, which leads to persistent activation of systemic inflammation. I feel like I'm going the way I don't want to go. I don't know how to move backwards on this. Okay, thank you. You just magically move backwards. Thank you, person in the back. Much appreciated. So how do we measure intestinal permeability? What we can do is we can give an animal an oral gavage. Think about it as an NG tube. You put something into the stomach. If barrier function is intact, what will happen? It will go into the stomach, the small intestine, the colon, and it will come out of our poop. If barrier function is dysregulated, it will go into the intestine. The intestine will become leaky, and it will become detectable in the blood. So this is what we do. We give an animal a oral gavage of a fluorescent tracer, something called FD4, and you look to see, is it present in the blood? If it's not present a lot in the blood, the barrier function is strong. If it's present a lot in the blood, the barrier function is weak. There's hyperpermeability. So we did this in mice. We did this in mice for the first couple of days after cegal ligation and puncture, starting as early as 6 hours. So you see that permeability is markedly abnormal, markedly worsened after the onset of sepsis. And association. Sepsis is associated with intestinal hyperpermeability. Is it model specific? For those who haven't heard me speak before, who here thinks about pneumonia and says, oh, it's the gut? Okay, a lot of you have heard me speak before. So why are you still here? So this is permeability following Pseudomonas pneumonia, having nothing whatsoever to do with the intestine. You give an animal Pseudomonas pneumonia, you see the exact same thing. The intestine becomes hyperpermeability because by definition, our patients who are dying of sepsis are dying of a systemic disease. Pneumonia is a systemic disease, intra-abdominal sepsis is a systemic disease, so it's almost a trick question. Obviously, you think about the lungs with pneumonia, but it's not just the lungs. Our patients who are dying of multi-system organ failure. It's affecting everything, including the gut where you see intestinal hyperpermeability. So again, we have an association. And we see various tight junctions are perturbed. It depends upon which model we're talking about. You talked about Claudin 3, and what we have found is that we see an increase in Claudin 2, a decrease in Claudin 5, a decrease in occludin, an increase in JAM, and an increase in ZO1. It doesn't really matter what, other than to say some of the tight junctions in the intestine are perturbed, some of them are not. What's the relevance of this? It gives us something that potentially we can intervene on to move from association to causation, a potential mechanism that allows us to say potentially 5 years, 10 years down the road, we can actually target this if this is important. So that leads to the question, what happens if we target permeability? So myosin light chain kinase, I don't want to get too nerdy sciencey in you guys, but MLCK, the bottom line, increases permeability. You're welcome to read all the basic science behind how it increases permeability, but it increases permeability. This takes you mechanistically through it. It phosphorylates, it initiates, it contracts, changes tight junction, and ultimately increases permeability. What happens if we knock it out? If we knock it out, you change every one of those. You don't phosphorylate, you don't initiate, you don't reorganize, you don't increase, and permeability should go down. Simplistically, we have a way to target permeability to see, is this association with intestinal hyperpermeability causative of mortality? So I already showed you this. This is what happens to a regular mouse if you make it, give it, I think this is cecal ligation and puncture. You see an increase in permeability. What happens when you knock out MLCK? Permeability totally normalizes. You don't see any increase at all. Now, knowing that we can normalize intestinal permeability, we can ask, is this mechanistically important? How? By looking at survival. First, we see, does it do anything else? Yeah, it changes inflammation. Change permeability, what happens? Change inflammation. This is systemic IL-10. And then can we say, does it change survival? In a major, major way. So we change intestinal permeability, we normalize it, and when we normalize permeability, we markedly improve survival. Few minutes left. Going to go back in time to what started our lab. The gut epithelium, the thing that's half the size of a badminton court, is actually a fairly complex thing. Cells are born at the base of the crypt. They migrate up to the top of the villus where they're exfoliated or die by apoptosis. The entire migration, the entire birth to migration to death in the intestine takes 3 to 5 days. We have an entire different intestine than we had last week, and then we're going to have next week. High turnover thing. Again, importantly, there's death. There's life, there's death, and they're balanced. People are born, people are die. We have a similar number of people. What happens if you change the ratio between birth and death? Long term, that's called cancer. Short term, what happens in the ICU? So short term, this is my mentor from 20 years ago, Richard Hotchkiss. This is short term in the ICU. Make an animal septic, cecal ligation and puncture. We see an increase in apoptosis, programmed cell death. This is work that we did looking at pneumonia. I'm not going to ask for a show of hands again who thinks about pneumonia, thinks about the gut. This is pneumonia, and we look for cell death in the intestine. We took the most common nosocomial gram-negative, Pseudomonas in the middle, the most common community-acquired gram-positive, pneumococcus on the right. Middle's published. We never got around to publishing the thing on the right, but it's the same thing. You see an increase in apoptosis. So once again, I've shown you an association. Sepsis induces increased cell death in the intestine. And this is in people. I said I wouldn't show any more people. In mice, I said I wouldn't show any more people data. This is people data, but it's autopsy data. Same thing. You look at autopsy data right after people die, and we see an increase in apoptosis, specifically in septic patients. And so now what happens if we prevent that? Because this will tell us whether an increase in apoptosis is good, bad, or indifferent, whether it's mechanistically important. So we take transgenic mice that overexpress the anti-apoptotic protein BCL2 explicitly and exclusively in the gut epithelium. Importantly, normally these mice are fine. Their intestines aren't bigger, they don't develop cancer, they live normally, everything is normal. So they have no baseline phenotype, but they prevent sepsis-induced apoptosis. That's showing you this. So on the left is a wild-type mouse that is septic, red is apoptosis. On the right is an animal that prevents apoptosis specifically in the gut epithelium, that's the blue. You see the absence of red. So we prevent gut cell death in the epithelium, and now we say, is this mechanistically important? We understand there's an association that it's increased. What does it mean? Here's our survival data. So we take this, we overexpress BCL2 in the gut, and we see a doubling of survival in intra-abdominal sepsis, and you say, but you're sticking a needle in the intestine, preventing gut cell death in the intestine. What happens if you do it in pneumonia? Again, who here thinks about pneumonia, thinks about the gut? I do. And so you give an animal pneumonia, you never touch its abdomen, you prevent gut cell death and only in the gut, and you improve mortality 10 times. So in conclusion, gut failure is common in the ICU. The microbiome, as our two great speakers spoke about before me, is converted to a pathobiome, and mechanistically clearly plays a role in mortality from sepsis. Intestinal permeability also mechanistically plays a role, and intestinal apoptosis also plays a role. A lot of work over a lot of years. I don't do any of the work. I think probably one person in my lab, some here is in the background. Zhang Feng, if you're here, good job. Acknowledges to many people who've done the work over time. Thank you.